Walking Stick Library presents Human-Animal Chimeras with your librarian hostess with the mostest, Eileen Kramer. And remember, you can catch this stream live on Twitch. Anyway, tonight we have another wonderful video, and this one was brought to you by Big D7615. And he says, this is on Cloud Hub. You're looking at Cloud Hub. You are in the belly of the beast. There is a big secret being hidden. Elites are not just trafficking, abusing, and sacrificing children, but also conducting disturbing experiments mixing human and animal DNA. Their dark agenda has been slowly revealed, with artists like Patricia Piscini supposedly exposing it through their work. Let's have a look-see.
Yesterday, the Senate passed the Endless Frontier Act, a bill that commits nearly $250 billion to promote emerging technologies so that America can keep pace with Chinese innovation. Back in May, Democrats shot down an important amendment outlawing U.S. participation in research that would create human-animal hybrids, also known as chimeras. China's already doing it, announcing in April that an international team successfully grew human monkey embryos and sustained them for 20 days in a lab. The only question is, what will they do next? Currently, the National Institutes of Health has banned U.S. funding for chimeric research, but that ban could very well be lifted. And who's to say that these agencies will play by the rules anyway? Remember, President Obama banned funding for gain-of-function research back in 2014, but New York AD found a way around it. They laundered, it's the NIAID group, and they laundered hundreds of thousands of dollars to the Wuhan Institute of Virology through a nonprofit group called the Eco Health Alliance. The rest is history. Chimeric research is a Pandora's box, that's obviously from hell, that should not be opened. But the scientific community, spurred on by their own hubris, may very well open it, using American taxpayer dollars. And if that happens, the Democrats will have blood on their hands for not joining the GOP and stopping it when they had a chance. Joining me now for reaction, I don't even believe, Laura Logan. Here we are. It's the 21st century. We're two very smart women. We have to talk about chimeras and how maybe the government might do it. You've been very involved in your research and stories and coverage on certainly COVID, what's been happening there. Give me your take on the seriousness of this. China's doing it, and what we have to be aware of.
You know, Tammy, one of the things that strikes me the most about this story is there's an old adage, be careful of what you don't know. And that has never been more true than with this subject. What exactly do we not know here about how far, what is the level of involvement the U.S. has had in any kind of research like this? And what else has the NIH and other divisions of the NIH been involved in? Because when it comes to Dr. Fauci and what we know about COVID, we're just beginning to find out, right? We're at the tip of the iceberg in terms of uncovering what was really going on with gain-of-function research. And the whole sort of smokescreen of the Wuhan lab really obscures the fact that Dr. Fauci and the NIH through NIAID had been funding gain-of-function research for years. And there's a lot of money that's missing here. And there's another U.S. agency that's missing from this equation. It's called DARPA, which is the Defense Research Agency. So what were DARPA and Dr. Fauci and the NIH doing with gain-of-function and various other programs like this one, which we know so...
I'm going to freeze it. It's time to do some research. Now, first thing we want to do is go over to, first thing we want to do is set out our topic. And I say chimeras, but it's chimera, apparently. So what we are going to do is that I'm not interested in animal chimeras. I'm interested in human chimeras. And I don't like where that is. Let's try that again, and let's change it. I wondered why that pen was so skinny. Some things you have to fix, and we'll fix all of them eventually. We're interested in chimeras that use humans. If somebody wanted to make a chimera with a horse and a donkey, I don't think we'd care. Or maybe a cow and a pig. I mean, it'd be interesting, I suppose. But, you know, or breed various fish together to get a nice hybrid. I don't think we're all that interested in that. So we want chimeras that are partly human. And it's the human part we care about. So again, I've got to set everything up because I always do, and I will check my chat once this is in place. So if you said something you haven't slipped off of the world. No, it's just Mio tonight. Human. And over here we have, and there is a second way to spell this, chimera, which I say chimera, and I'll probably slip back to that. Because it looks like chimera. And we're not going to go there. We're going to go here. A and D. And we want what's between these, and we're going to go to Medline, and we're going to treat this Medline search just like any other garbagey search that we commonly do. We do lots of garbagey searches. This is just a garbagey keyword search in Medline. The trick here is going to be a lot of close reading. So what we're going to do is get this guy over here, make him into Google, because I can do that the back way. We'll go in the back way. Here's Google. Get rid of that, and we're going to go to GSU Library. You can do this in any academic library. This is a fairly common database. Whether you'll get the full text for everything is another story. We skip this box. We want all that wonderful Medline-y goodness. So we go in, and again, we want human and chimera with an asterisk. We're going to scroll down in the databases till we hit Medline with full text and let ourselves in. And we have to change this. I changed my password today. That should work. Yes, save it. I may get duoed. No, I didn't get duoed. I get passed through. Here we go. Here we go. Come on. Baby wants a pair of shoes, and baby got a pair of shoes. And all we do is type this in. And remember, an and can go in any order. An and can go in any order because it's this guy. And if you're from Singapore, or if you had the new math, that's what we're doing. This is much older than AI. It works because it works. It's actually a kludge, but we like it anyway. And, okay, humans and mice. Mouse embryo chimeras, embryonal carcinoma stem cells, reflections on the winding road to gene manipulation. Okay, so they're making embryonal carcinoma as a stable cell line here. And again, remember, the human cells are getting by doing a tissue scrape in someone's mouth or a blood test from a volunteer. They're not taking any dead babies for this. Emerging human pluripotent stem cell-based human and animal brain chimeras, advancing disease modeling and cell therapy for neurological disorders. Okay. Debates on the humanization of animal brain chimeras. Human-animal brain chimeras. Are we putting the cart before the horse? Okay, I like that this is a 2024. This wasn't the way I went on walkthrough, but I don't have to do it the way I went on walkthrough. This is in the Neuroscience Bulletin. It is September 2024, so it's about 6 months old. Let's click this, find it, and let's hope we don't go clunk. Because it's fun to go in and not go clunk. That didn't work very well. Let's try again. Okay. And I think we can log in. I think we've got access to Springer. That's not it. Ouch. There's another way to get in. Log in via an institution. This is what we call being persistent as hell. That's not going to work. We're going to try it again. Give it another break. That didn't take correctly. That is correct. And we can't get in. But we can get to the abstract. And this may tell us a little more. Human pluripotent stem cells models provide unprecedented opportunities to study human neurological disorders by recapitulating human-specific disease mechanisms, in particular HPSC, human pluripotent stem cells, which are often made from just tissue samples. Based human-animal brain chimeras enable the study of human cell pathophysiology in vivo. Okay, without getting somebody sick, in chimeric brains, human neural and immune cells can maintain human-specific features, undergo maturation, and functionally integrate into host brains. Okay, allowing scientists to study how human cells impact neural circuits and animal behaviors. Okay. So we have live rats here. The emerging human-animal brain chimeras hold promise for modeling human brain cells and their interaction in health and disease, elucidating the disease mechanism from molecular and cellular to circuit and behavioral levels, and testing the efficacy of cell therapy interventions. Here we discuss recent advances in the generation and applications of using human-animal chimeric brain models for the study of neurological disorders, including disease modeling and cell therapy. Now, whether anybody's done this, we don't know. These are some of the pictures. Human cerebral organoids, a new search for human applications of, okay, I don't see anything about making more chimeras. So this is as close as we're going to get here. We can pick some other ones up. Debates on the humanization of human-animal brain chimeras. Are we putting the cart before? I just said I knew I would do that. And again, let's see. Now, a lot of the brain models in animals are terrible. That's one of the reasons we do this, and once again, we're not having any luck. This is that same, this is medicine, healthcare, and philosophy. Let's see if we can get in at Georgia State University. And we couldn't get in. Okay, research on human-animal chimeras have elicited alarms and prompted debates, those involving the generation of chimeric brains in which human brain cells become anatomically and functionally intertwined with their animal counterparts. This means you can do brain research that would not be ethical in humans but is ethical in a rat or a mouse in varying ratios, either via xenografts or embryonic co-development, have been considered the most problematic. The moral issues stem from the potential for humanization of the animal brain, as well as speculative changes to the host animal's consciousness or sentience, with consequential alteration in the animal host's moral status. So they're worried about the rats. And I'm assuming they're using rats and mice, and I always think rats because rats are bigger. But there could be mice too. However, critical background knowledge appears to be missing to resolve these debates. Firstly, there is no consensus on animal sentience vis-a-vis that of humans, and no established methodology that would allow a wholesome and objective assessment of changes in animal sentience resulting from introduction of human brain cells. Animals are already sentient. Mice feel pain, rats feel pain. They know pleasure, rats joy jump when they play the right kind of games. Knowledge in interspecies comparative neuropsychology that could allow effective demarcation of the state of humanization is also lacking. Secondly, a moral status as a philosophical construct has no scientific objective points of reference. Either changes in sentience or humanization effects would remain unclear unless there are some neuroscientific research grounding for a bioethical stance based on moral status of human-animal brain chimeras to map meaningful contributions to regulatory policies. It might first need to adequately be informed by and with arguments constructed in a manner that are factually in line with science. It may be prudent for approved research projects, including the generation of human-animal brain chimeras, to have a mandatory component assessing plausible changes in sentience. So, if the animals get too smart for their own good, we got a problem. Okay. Here we have digital polymerase chain reaction for quantification of hematopoietic chimerism, which means making blood cells by insertion deletion of personalized selection for different chimerism status. And here we have toward developing human organs via embryo models and chimeras. And this is 2024, June 20th. Let's see if we find it. Now, before we'll take a look and then we'll do another look. Let's see how we do. Don't tell me we got one. We do got one. We do have one, and this is cool. Very, very, very cool. Okay, embiggen, get rid of the thing on the side. Let's have a look-see, and this is your summary. Developing functional organs from stem cells remains a challenging goal in regenerative medicine. Existing methodologies such as tissue engineering, bioprinting, and organoids only offer partial solutions. This perspective focuses on two promising approaches emerging from engineering human organs from stem cells: stem cell-based embryo models and interspecies organopoiesis. Both approaches exploit the premise of guiding stem cells to mimic natural development. We begin by summarizing what is known about early development as a blueprint for recapitulating organogenesis in both embryo models and interspecies chimeras. The latest advances in both fields are discussed before highlighting the technological and knowledge gaps to be addressed before the goal of developing human organs could be achieved using two approaches. We conclude by discussing challenges facing embryo modeling and interspecies organogenesis and outlining future prospects for advancing both fields toward the generation of human tissues and organs for basic research and translational applications. Okay. I'm going to check our house. Did anybody come in to enjoy the white noise? No. Okay, this is interesting, and it's getting a lot closer to what we want. What I did was just open the record. This is the title. This is the link. We get down here. We have chimera as a, as a subject heading. I can make this work a lot better. Off to the mesh headings we go. I type in chimera. Now, we want to look at their subject headings, their scope note before we go further. An individual that contains cell populations derived from different zygotes. It's that simple. We also have transplantation chimera. And again, I can click this, and what does it say? An organism that, as a result of transplantation or donor tissue cells, consists of 2 or more cell lines descended from at least 2 zygotes, the state may result in the induction of donor-specific transplantation tolerance. I'm going to go back the way we came, and we're going to pick chimera. We're going to make them a major concept, and we're going to let it rip. You'll see what we do next. Now, we could add human back in, but I've got something else in mind. I remember that until very recently, there was a woman who, she's still alive, she's on dialysis, a woman who received a humanized pig kidney. Now, those piggies are chimeras, and pigs make excellent xenodonors. They can't complain about malpractice. They're pork chops, baby. Pigs. Actually, better than this, we have 2 words for pigs. Swine. And we're going to put an or between this. So this is going to get very fancy. And this becomes an alternate. We're interested in humanized organs made from pigs. This is a fun way to look at chimera. But we also looked at, how you could make brains that you could test in mice. And what we're looking for is chimera, actually as a subject heading, and pigs or swine. And this is our binky bink bink. This is our, get my pen back. This, X, and in here we have Y. I'm going back to Y letters here. Bold. I like to make this a different color so that we know we've got different kind of operators. And or, that should be round on the bottom. That's union, Z. And the little nest, parentheses is called the nest. The nest can be on either side of the intersection or the and. This is a commutative search. Inside the parentheses, it's also commutative. As long as you leave the parentheses around Y or Z, you're fine. Okay, folks, let's add it in. And pigs or swine. In fact, pig or swine. Let's see what we get. And we're going back to 1962. I think we only want the last 5 years. There's only 12. Okay, here's one right here. Domesticated cynomologous monkey embryonic stem cells allow generation of neonatal interspecies chimeric pigs. And again, this is done in China. It's in Protein and Cell. It is 2020, and we can try to find it. And they don't want to do it with humans for some reason, so they're trying the technique out with monkeys. They have much more primate research in China than we do. And here's your abstract. Blastocyst complementation by pluripotent stem cell injection is believed to be the most promising method to generate xenogenic organs. Xeno means other. I think that's xenogenetic. I think they left out the T. Xeno means other. Xenophobia is fear of the other, the foreigner. However, ethical issues prevent the study of human chimeras in the late embryonic stage of development. Primate embryonic cells, which have similar pluripotency to human embryonic stem cells, are a good model for studying interspecies chimerism and organ generation. However, whether primate embryonic stem cells can be used in xenogenous grafts remains unclear. In this study, we evaluated the chimeric ability of cytomolgus monkey Macacia fascicularis, it's a macaque, embryonic stem cells in pigs, which are excellent hosts because of their many similarities to humans. We eat the same diet, or they're a little bigger than we are, although some aren't. There are potbellies that get to be maybe 100 pounds. They're a little bigger than we are, but they're close to the same size and eat the same diet. We report an optimized culture medium that enhanced the anti-apoptotic, which means apoptosis is cell death. So these cells were good at staying alive. Anti-apoptotic ability of cytomegalovirus embryonic stem cells and improved the development of chimeric embryos, in which domesticated cytomegalovirus embryonic stem cells were injected into pig blastocysts, differentiated into cells of 3 germ layers. In addition, we obtained 2 neonatal interspecies chimeras. So they put them back in the piggy, in which we observed tissue-specific DESC differentiation. Taken together, the results demonstrate the capability of DESCs to integrate and differentiate into functional cells in a porcine model with a chimeric ratio of 0.001 to 0.0001 in different neonate tissues. We believe this work will facilitate future developments in xenogenic organogenesis, bringing us one step closer to producing tissue-specific functional cells and organs in large animal model through interspecies blastocyst complementation. Okay, I want all of you to see the number up here. These are only a tiny bit monkey. You're not seeing a pig eating bananas, a pig with a and these are old monkeys, no prehensile tails. You're not going to see a pig with prehensile hands eating bananas. Not going to happen. And they do it with interspecies blastocyst complementation. I'm going to put that down here in a corner. Interspecies blastocyst, which is a hollow ball of cells. It's a very early embryonic stage. They had AP Bio. That's what we're looking at. And it doesn't like any of my words, and that's just too stinking bad. Let's go back to 100%, folks. So that's another way to do this search. That's helpful. And here we have more. Immunization with recombinant fusion of porcine reproductive and respiratory syndrome virus, modified GP6 ferritin elicits enhanced protective immunity of pigs. That's a different kind of chimera. And here we have again, in vitro and in vivo interspecies chimera assay using early pig embryos. And again, this is 2020, came out during COVID. They've been doing this for a while because the goal is to get organ transplants. Okay, let's see if we can get in. Spinny, spinny. We clunked. Okay. Durable mixed chimerism may permit a subsequent immunosuppression-free intestinal transplantation, a proof of principle study. Wow, let's see if we can get in. And again, everybody loves to do swine. If I were a student, I'd be ordering a lot of interlibrary loans tonight. Okay. And we can try to get access, but right now this is as good as we get. Okay, intestinal transplantation is the definitive treatment for intestinal failure, but has high rejection rates among solid organ transplants. Usually you end up with some kind of ostomy, which is pretty gross. My grandmother had one. So intestinal transplantation is the definitive treatment for intestinal failure, but has the highest rejection rate among all solid organ transplants, requiring high doses of immunosuppressive medication, which is associated with high rates of infection, graft versus host disease, and malignancy. Transplant tolerance would overcome the need for long-term immunosuppression using non-myeloablative conditioning. Our laboratory has developed a novel swine model for hematopoietic stem cell transplantation that produces durable mixed chimerism and immune tolerance without toxicity. Okay, non-myeloablative conditioning, I don't know what that is, but I know ablative means scraping, myelo means muscle, and non means not. So we do this without scraping muscles. We've developed a swine model of a hematopoietic stem cell. That is a stem cell that can turn into different kinds of blood cells. We have some of those in our bones right now. Of hematopoietic stem cell transplantation that produces durable mixed chimerism and immune tolerance without toxicity. We investigated whether durable mixed chimerism would promote tolerance of subsequently transplanted donor-matched intestinal allografts without immunosuppression, using miniature swine with a defined major histocompatibility complex. We performed HSCT, hematopoietic stem cell hematopoietic stem cell transplantation across an MHC, which is a class 1 halotype mismatch. Okay, so we're going to put these chimeric stem cells into somebody with a different halotype, which is sort of a guaranteed rejection. And we did this with miniature swine. We're not using humans. We're not using monkeys. Immunosuppressive therapy was stopped by day 45. And mixed chimerism was evaluated using flow cytometry, and mixed lymphocyte reaction assays were used to evaluate cellular responses. Subsequently, orthoptic ITX was performed. And I don't know what that is, but I think it's intestinal surgery without immunosuppressive medications, using a donor that was MHC matched to the HCST donor. The recipients were observed for 4 weeks and euthanized for tissue collection and mechanistic assays. After the HCST, the recipients developed durable multilineage chimeric cells and apparent deletional tolerance. After the intestinal surgery, the recipient showed no clinical or histological signs of rejection, and chimerism was unchanged. These results demonstrate the potential value of generating durable, multi-species, durable chimerism to achieve transplant tolerance. So what they would do is a stem cell transplant, putting in the new species stem cells, and this would create the mixed chimerism that enables the piggy to handle an intestinal transplant. And piggy guts are a good stand-in for human guts, because piggies eat the same diet we do. Poor piggies. Well, piggies end up being pork chops and raised in factory farms anyway. And here we have the ethics of creating and using human-animal chimeras. And we can, let's open this up in a new tab and see how we do. We hang in there. And it looks like we might be able to get the whole thing. Let's try it. We got in. Oh, sweetness. Sweetness, sweetness. Let's get the PDF because we can embiggen it. Get the whole page to ourselves and embiggen it. Rapid advances in gene editing and stem cell technology have expanded the range of possible future applications in human-animal chimera research. Most notably, recent developments may allow researchers to generate whole personalized human organs in pigs for the purpose of transplantation into human patients. Though human-animal chimera research in small animals such as mice is routine, human-animal chimeric research techniques are now increasingly being applied to larger animals. Moreover, these chimeras include increasing amounts of human material, which is potentially present in more morally significant locations, such as the brain and the reproductive system. These developments raise important ethical questions about whether we should create chimeras, and if so, how we should treat them. Answers to these ethical questions are needed to inform the development of policies regulating human-animal chimera research and its application. Here we provide a review to some of the most important and widespread ethical concerns. Well, this looks good, and this is 2020 again. I don't know why this all disappears 5 years ago. The other thing that's kind of interesting down here is hybrid origin of a primate. Can we have a look? Hey, nature, you're wonderful. And here we have somebody doing chimeric-inspired art, just like in that video, which we didn't see the end of. Let's see what's happening here. These always have to be made very big. Okay. Golden snub-nosed monkey, black and white snub-nosed monkey. Somewhere down here we got gray ones. Hybridization is increasingly recognized as an important evolutionary force for generating species and phenotypic diversity in plants and animals. This is especially common in lineages that can tolerate whole genome duplication and in increased levels of ploidy. However, the role of hybridization in generating species and phenotypic diversity of lineages without polyploidization is underappreciated, especially in non-hominoid animals. The snub-nosed monkey genus Rhinopithecus comprises 5 allopatric and morphologically differentiated species: the black-white snub-nosed monkey, the black snub-nosed monkey, the golden snub-nosed monkey, and the gray snub-nosed monkey, and the Tonkin snub-nosed monkey. These are all Asian monkeys. They possess the same chromosome number, and it has been speculated that they have hybridized in the past. To examine the speciation histories of these species, we generated a chromosome-level, high-quality reference genome assembly for the black-white snub-nosed monkey and analyzed 106 resequenced genomes for individuals of all 5 subspecies. We continued to multiple population genomic analyses, including admixture, d-statistics, phylogenetic reconstruction, and evolutionary scenario simulations to investigate the genomic admixture of these species. We further applied genomic selective scans and functional assays to reveal the likely genetic basis of mosaic coat coloration of the hybrid species. Possible mechanisms of pre-mating and post-mating reproductive isolation barriers between hybrid species and its parents are briefly discussed. So, sometimes it happens in the wild. And I have to say, I'm going to come back inside for a moment. I have to say, and I'm going to say it, that if this ever matures and if Todd or his relatives need an organ, they're going to go first in line for that chimeric organ, if that's what's on offer. And I'm going to see if anybody came in. I hope you all enjoyed this. Thank you so much for watching. And don't forget, you can catch me live on Twitch.